Hi everyone, my name is Gurleen and today I'm going to present a poster on the Wilson's disease. As we all hear this term, whenever we hear this term, the question comes to our mind, what is Wilson's disease? So it is a genetic disorder that allow copper to build up in the body and cause damage. The question is what kind of damage, how, and why? To work well, the body needs a small amount of copper. Copper is a metallic element. It helps keep the immune system healthy, build strong bones, and form red blood cells. We absorb copper from a variety of foods such as nuts, seeds, oysters, and liver. Normally, our bodies regularly dispose of copper to prevent it from accumulating in the body. Some children are unable to get rid of the copper. The inability is a disorder that is known as Wilson's disease. The excess copper is stored in the main organs such as brain, liver, kidneys, and eyes. In the liver, extra copper causes damage and scarring. This causes the liver to stop working correctly. There are some signs and symptoms of Wilson disease. They're more likely to be in 10-year-old children and the people who are more than 10 years. In the Wilson's disease, copper usually accumulates in the liver and brain. So this is why the symptoms are mainly liver disease and neurological problems. The symptoms include the tiredness, yellow skin, that's jaundice, or yellow color of the white of the eyes, the cornea, the enlargement of the abdomen, vomiting blood. The symptoms of the neurological problems are confusion, emotional or behavior changes such as depression and anxiety, and then slow or decreased movement, slurred speech, tremors in the hand, and clumsiness, worsening of the academic performance, which results because of the above mentioned problems. A change or mutation in gene causes Wilson disease. Children are born with it. It is a autosomal recessive disease, which is also known as hepatolenticular degeneration. There is an history related to this. In 1854, Friedrich was able to observe it. And then later on in 1912, this disease was subsequently named, which was named after Samuel Alexander Kinnear Wilson. The genetic basis of Wilson disease was elucidated in 1990s when the gene was identified and mutations in the gene were linked to the disorder. Then what is the gene which is responsible for Wilson's disease is gene ATP7B. It is located on the chromosome number 13, which functions to encode a copper transporting P-type ATPase, which plays a crucial role in regulation of copper levels in the body. It is involved in the transport of copper into bile and incorporation of copper into ceruloplasmin, a protein that carries copper in the blood. The structure has several domains, which include transmembrane domains that facilitate copper transport, ATP binding domains that provide energy for the transport process, and metal binding domains that bind copper. The mutations can lead to Wilson's disease, a disorder which is characterized by excessive accumulation of copper in the liver and brain that we discussed earlier. Nonsense mutation, frameshift mutations, splice site mutations are some of the kinds which can occur on the chromosome number 13, which can result into the mutations as we are discussing about it. Genetic testing for this mutation is used in diagnosis. And then identifying specific mutation can help us in predicting the clinical course of the disease, and we can tailor the treatment strategies and so on. Research is ongoing to develop therapies that target the gene or the protein synthesis and the pathways to treat it. There are diagnosis and treatment. First of all, we have to find how we can diagnose it. What are the different ways we can understand or we get to know that this is the disease the patient is going through. First, diagnosis include blood test. To help confirm the diagnosis, doctor takes sample of the blood. So it tell us the amount of copper in the blood, the presence of liver enzymes. High level of these enzymes means the liver cells are damaged. The amount of a liver protein called albumin. Albumin level decrease in blood when liver is damaged. How long it takes for the blood to clot. The amount of protein which transports copper in the blood called ceruloplasmin. As we discussed earlier, its level are lower in Wilson disease. Other than that, liver biopsy can be done. in which doctor takes a sample of the liver. They check for specific changes in the liver. They also measure the amount of copper in the liver. Genetic test for Wilson disease. The doctor may run a genetic test, and we have to wait several weeks before knowing if the disease is there or no, because since it is an autosomal recessive disorder, a genetic disease which passes on from the parents to the children. There are several treatments for this disease. The very first includes chelation therapy. In this, we use some medications to remove the excess copper. The second one is zinc therapy, where we administer zinc acetate to block the copper absorption. Then we can have some dietary modifications in which we can limit the intake of copper-rich food like shellfish, nuts, liver, and then liver transplant could be another option in which in case the liver is severely damaged or it has been failed, we can transplant the liver. And regular monitoring, regular blood test can be run, and urine test can be run to measure the copper levels in the body. Other than this, in the conclusion, I would like to say that Wilson's disease is a rare disease, but it is a significant genetic disorder because it highlights the critical role of copper in the copper metabolism in human health. Through our understanding of the ATP7B gene and its impact on copper transport within the body, we all are able to gain insight into the molecular mechanism that lead to the pathological accumulation of copper in vital organs. The advancements in the genetic testing and the development of effective treatments such as chelation therapy and dietary management have greatly improved the prognosis for individuals diagnosed with this condition. Moreover, this journey into the world of Wilson's disease underscores the importance of ethical considerations in genetical science. The principles, the informed consent, the non-discrimination, the equitable access to treatment are of paramount importance in ensuring that the benefits of genetic research are realized in a manner that respects and protects the well-being of individuals and society as a whole. As we all continue to explore the genetic basis of all the other diseases as well, we develop innovative therapies. Let us remain committed to holding these ethical standards. Fostering a future where genetic science not only provides solution to health problems, but also promotes a just and a compassionate society. Thank you for your attention, and I hope this presentation has shed some light on the fascinating and complex world of Wilson's disease and the broader implications of the genetic science in healthcare. At the end, I would like to include the references, the articles, and the information I used to accumulate all of these at one place. Thank you so much.